Aha, there we go. Now we're recording. Okay. So, and actually, let me see, who do we have on board here? Okay, looks like, okay, Dana and Mike, you're on. Internally, we've got Georgina. Okay, anyway, so this is Bill Gillespie. I'll be teaching a portion of the course, and I'll be doing that along with Jonathan French, who's also here in the room with me today. And let me bring this up full screen. Okay, so in fact today I'll sort of introduce some of the first course material, probably for probably somewhat less than an hour here, and then Jonathan will take over. So this is our MI260 course, Model-Based Meta-Analysis to Support Decision-Making in Clinical Drug Development. Just to introduce you to some of the logistics for the course. So the intention of the course is to give you an introduction to model-based meta-analysis, primarily of summary data, but also to be able to analyze combinations of summary and individual data that come from clinical trials, and to use the resulting analysis to support decision-making in clinical drug development. The course duration and content is approximately equivalent to a single semester 3-credit-hour course in a typical institution of higher learning. And each week's topic here will be consist of a lecture on for about 2 hours, followed by a hands-on lab, typically about an hour. We'll be holding lectures on Wednesdays at 2 Eastern Time, and the labs will be on Mondays at 2, also at 2 Eastern Time. For those of you who want to receive a certificate for the course, this slide talks about the expectations and requirements to obtain that certificate. All students are expected to attempt our hands-on exercises prior to the lab session, though we'll not actually be grading those homework assignments. That's basically your opportunity to learn how to implement the methods we'll be talking about. And of course, you'll need that knowledge to complete some of the other course requirements that will be graded. We will review the solutions to the assigned exercises, and that's something we do primarily during that hands-on lab session every week. There'll be a midterm take-home exam assigned at the midterm of the course, then a final take-home exam at the end of the course. Let's see, we're saying that will be due a week after it's posted. Students will also be required to complete and submit a modeling project before the end of the course. The idea is to come up with a project based upon a real world or something similar to a real world problem. And it's come up with a problem that will include various elements like data assembly, model development, and model evaluation, and perhaps also some model simulation. And then there'll be a brief report that will be used to base the grading on. And we'll give you some more details later on for that. So the course grade then is going to be based on a combination of those, and here you can say I've got it down as midterm counts 25%, final another 25%, and then the project 50%. There is associated with the course a website, labeled here as a course content management site. You should all already have access to that website. In fact, that's probably how you set up your enrollment. And you can see there's the link is cited here. Postings to that site as part of the forums within that site will automatically generate an email message, to your email accounts. And that's where we will be distributing various course resources, like these slides, for example, as well as other course content. In addition, you have forums there, one labeled as a news forum, where is usually how we'll send out information to you about the class schedule, any assignments, and so on. Then there's a discussion forum, which is the best place for you to direct any questions, other than during the course time. You can use the forum then to direct questions to us in here, and and we can reply to it. In addition, other students in the class can also reply. There's a link to the GoToWebinar webcast registration form, but you've already done that, so that's not really important at this point. As I say, the course materials are also distributed there. Maybe it's worthwhile. Let's actually take a quick peek at that. Let's see, let me fire it up. Okay. Sorry, let me shrink the thing I've got on my screen. There we go. Okay, so here is the course website. Actually, this is the overall course website. Oh, I just realized I'm not going to know how to log into this, am I? I'm not on my own machine here, so let's see. What will it show me, if anything? Probably nothing. Excuse me, you want to toss me my computer? I would like to show it to him, but I don't remember my password. Let me take a look. Okay, there we go. Okay, now we're in there. Okay, so this is specifically the page for MI260, and if you haven't already been there, you can see again we've got the forums where you can go ahead and either read postings that are already there. So, for example, if I go into the news forum there, you can see things that you've probably already seen. For example, I had posted the query about the times. And you can go to the discussion forum where you can launch. You can go ahead, if you click on this, add a new discussion topic, you can go ahead and launch your own topic to direct questions to us. You can see on here the course handout, which is essentially just a 2-slide per page version of the slides we're looking at today. And then we'll talk about this other one in just a moment here. In fact, I think that's my next slide. This zip file here, MI260 USB.zip, contains all of the course materials that we put together so far in here. So you'll want to download that. Be prepared to wait a little while. It's about 80 megabytes. So depending upon the download speed you have available to you. As we go along through the course, you can see this is broken out by week in here. So anytime we add new content or want to mention any new elements in here, we'll go ahead and add them in to the various weeks as appropriate. Chances are we'll probably be changing the slides and as we go along, as we come up with either new content or choose to change some. So periodically you'll probably see the course handout offered again here because we've changed something. Okay, let's find the slides again. Okay. To do the course exercises in here, the assumption we've made is you'll be using your own computer. And the way we've dealt with this, since I realize a number of people may be working for companies that put various shackles on their computer to make it hard or impossible to install new software. So rather than require that you install software on those machines, what we've done is we've set it up in such a way that you can run run all of the course examples and all of the software from a flash drive. So you never actually have to do an installation. You simply have to be able to access the contents of that flash drive. So normally that will not cause problems with most companies' security issues here, since generally all of the work is done right on there. All of both any source files as well as any results from the analyses are generally kept there. So the way we've got the course materials, again, they're in that zip file here called MI260 USB.zip. To use that, what you want to do is first download it to whatever is appropriate. Go ahead and unzip that file. And when you unzip that, it's going to create a directory called MI260 USB. Now, go ahead and get a flash drive that has a capacity of at least 1 gigabyte and copy the contents of that directory, that MI260 USB directory, to the flash drive. And just a reminder here, copy the contents of that folder, not the folder itself. Once you do that, I can actually show you the basic files here. Or actually can. I just realized I'm not on my computer. Actually, I probably can't. We won't go there today since I don't have that. We'll go over that on Monday in more detail. In fact, just so you know, we won't be assigning a hands-on exercise today, but on Monday for the lab session, I'll basically go over the use of the use of the various course materials here, or at least the beginning of it, to make sure that you're able to run R and run WinBUGS from the installation. So we'll go over that on Monday. So let's start digging in a little further. Okay, so I sort of mentioned the objective already. I want to provide an introduction to model-based meta-analysis. The intended audience here are people who have some PK/PD or statistical modeling experience and already have some familiarity and hands-on experience with nonlinear regression, mixed effects model, and Bayesian model using WinBUGS and are already familiar with using R. So today this is the course outline. We're already going through the introduction now. We'll spend a little time today sort of describing the motivation behind model-based meta-analysis in clinical drug development. And I'll give you a few what I've termed motivating examples. And then at that point, Jonathan's going to be taking over and talking about some general items here, things like systematic review and planning a meta-analysis, and I'm talking in terms of what we might term traditional meta-analysis methods in here. Many of the things on this page sort of are follow-ons to that. So initially talk about fixed effects meta-analysis and then get into random effects meta-analysis and meta-regression, talking about some topics that crop up with meta-analysis, particularly when we're drawing on published data, things like selection bias, dealing with missing data, combining various categories of data, and then finally some network meta-analysis. Then we'll dig into what we've tended to label as model-based meta-analysis, and we'll talk about, initially talking about dealing with cases where we're dealing with sample mean data, perhaps augmented by sample standard deviations. Talk about exercising the resulting models via population simulations, and then talk further about some issues that arise when using summary data in here, dealing with issues that come up with the fact that most of us in pharmacometrics tend to think in terms of modeling individual data, and how do we model things like time courses and dose response within each individual patient, but and we develop models in that context. Well, what do you do when you want to apply those models in the context where you do not have individual data, and what are the implications? some issues that arise in analyzing longitudinal data. Then we'll talk about how do you model other types of summary statistics besides things like sample means. Things like fraction of patients that have some particular outcome, fraction of patients at each level of some ordinal scale, number of events per patient, and summary statistics for time to event measurements. And then a few other issues here and there. I suspect that I should probably mention this original course outline was developed in the context of our 3-day workshop on this topic. We're likely to do some revisions as we start getting out into this part of the course, maybe adding some new material and some additional hands-on examples as we go into some of those later topics here. And where we go, and I think I'll jump right by that. Okay, so let's get into sort of the motivation behind this. In particular, why do it? In particular, why do meta-analysis and model-based meta-analysis in particular? And as I argue here, the primary rationale for model-based meta-analysis is to improve decision-making by better leveraging prior information that comes from multiple sources. Often those multiple sources being, various publications, but they may also be other elements that may come from proprietary sources, for example. I mentioned here, decision makers generally attempt to consider that kind of prior information, but it tends to be done in a relatively qualitative manner. In addition, there may be a selective element in it due to the fact that each individual decision maker is usually aware of only a subset of that prior information. Model-based meta-analysis is trying to make this more rigorous, in particular trying to make that process more quantitative and comprehensive. I also argue here that it's also desirable to do this in partnership with those decision makers, where in that process you try and make clear, well, as I say here, you want the process and results of that meta-analysis to be made visible or more transparent to those decision makers. By that, I don't mean you necessarily want to show them every ingredient and step in making the sausage, if you like, but you definitely want them to be part of the process of evaluating assumptions that are being made, and probably how you're viewing the source information for building those models, and where appropriate, drawing on their expertise on interpreting that information. Anyway, the end result of that partnership is that those decision makers are better informed, and they can contribute their knowledge to the modeling process, leading to better, more trusted models and better model-based inferences. Of course, out of that, hopefully better decisions overall. Continuing on here, part of the idea here again is you want to leverage prior knowledge from multiple sources. Those may be multiple sources of the same kind, or maybe of heterogeneous types. You may want to draw on data for your new chemical entity of interest that may come from preclinical sources, maybe phase 1 safety and biomarkers, as well as clinical safety and efficacy studies. You may also draw on information about the target disease and affected physiologic systems that go outside the clinical trial context. And importantly, you want to draw on knowledge and data about related compounds. And this could come from proprietary public sources. It may be data relating to analogs that share the same mechanism of action and perhaps some chemical properties as the NCE of interest. Also, you generally want to bring in data for competitors because that data may serve as benchmarks for comparison. Reasons for doing this include, of course, comparison with competing drugs and treatments. Another reason for bringing in information about some of those other drugs and treatments is particularly if you have information about analogous compounds, that may permit a certain degree of information sharing in such a way that you might be able to share some model parameters across different compounds. And if that's the case, those shared model parameters are more precisely estimated, and the result of that then is that your comparative inferences are also more precise. In some instances, it may be possible to leverage model-based meta-analysis to actually make predictions about clinical outcomes that have not been observed for the new compound that you're developing. So, for example, in some cases, you may be able to build models for bridging from preclinical to clinical or from biomarker to clinical outcomes based upon data from other drug development programs, but then use that in conjunction with models for those preclinical or biomarker outcomes to be able to make inferences about what might happen with that new chemical entity, even though you've yet to obtain that kind of clinical data for that drug. And what sort of decisions might benefit from model-based meta-analysis? Well, one, model-based meta-analysis can enhance dose selection and proof of concept decisions in a couple of different ways. For example, model-based meta-analysis can improve quantitative comparisons with competing treatments, and that may permit better selection of a dosage regimen that would perform as well as or better than those competing treatments, and thus be a good basis for dose selection. Alternatively, those modeling results may actually demonstrate that no dosage regimen for your new drug compares favorably relative to the comparators, and in those cases, it may provide you a basis for making a more precise and an earlier decision to terminate development and move your resources to where they're better used. Did I jump? Yes, skip the slide. Sorry. There we go. Okay, yeah, this is just commenting that in some cases, models developed by model-based meta-analysis may also be suitable for simulating clinical trials and therefore improving decisions regarding the design of such trials. And I comment here that it can be particularly valuable in cases where no clinical efficacy data is yet available for a given new treatment, and in cases, like I mentioned before, where you might be able to use some bridging model based upon other data to make inferences about what those outcomes might be and do trial simulations based upon those. And I think I'll skip the next line there. And I was just commenting that in some cases it may also be useful to use use model-based meta-analysis for decisions related to market differentiation and perhaps even choice or prioritization of indications. This was just to give you some illustration of some of the uses of model-based simulation that have been reported, and actually what you're looking at here is a table from an article that Brian Corrigan did a few years ago that illustrated the increasing use of model-based meta-analysis. Well, this is about 5.5 years ago, and thus it's a bit long in the tooth. If you actually take a look now, both in published literature as well as things like abstracts and things like Page and ACOP, you'll see that the list is much longer at this point. So what I wanted to do over the next little bit of time is spend some time going over a small number of examples just to illustrate the use. But actually before that, let me take a quick breather here. I haven't actually looked to see if anybody posted any written queries here. Maybe I should do that real quick just to make sure I haven't. And where to go? Oh, that's right, I shrunk it. Okay, nobody's put anything on there, but let me go ahead and take a breather for a quick second, see if there's any questions about the various elements of the course or any of the things I've talked about so far. Okay, nothing so far, so assuming nobody's been talking while they've had their mute button on, I guess I'll go ahead and move on. Okay, well, let me go ahead and present a few examples that just illustrate the use of model-based meta-analysis. This is one that Yatmandama and some co-workers had done a number of years ago. You can see it was published in 2005. It's sort of, I guess it was one that I would argue is probably the one that sort of kicked off much of the interest within the pharmacometrics community. They were looking at the development of gemcabene, as it says, a lipid-altering agent, primarily looking at it for its ability to lower LDL cholesterol. And meta-analysis was used to do things like look at dose response, try and optimize dose, well, and in the end, you'll see it also contributed to maybe even more key strategic decision. So in this article, they did a meta-analysis of mean LDL cholesterol percent change from baseline based upon results from 25 clinical trials, 13 of which were just looking at statin monotherapy. Then there were 4 trials of ezetimibe monotherapy, some trials looking at combinations of statin and ezetimibe. Let's see, 3 trials of just gemcabene monotherapy, and then finally a trial that looked at statin gemcabene combinations in here. I guess the one comment here is when gemcabene's development program was launched, ezetimibe was not on the market and had not been a key comparator at that point, but it had been launched during the course of the development of gemcabene. Just comment here, the original work on this also included modeling of a number of other endpoints that were of some interest here too, but in this case, the key decisions were based upon the LDL outcomes. Here we're looking at the results of that model-based meta-analysis, looking at statin dose response, and in this case we're looking at statin monotherapy. So here we're looking at on the y-axis it's LDL percent change from baseline, x-axis is the statin dose. So for example, on the upper left-hand corner where we've got atorvastatin, the upper curve here is the atorvastatin monotherapy, and the lower curve describes the LDL percent change from baseline for atorvastatin plus 10 milligrams of ezetimibe. And you can see the same sort of pattern across simvastatin, pravastatin, and lovastatin, showing roughly the same sort of incremental benefit of taking ezetimibe on top of the statin, and an incremental benefit that's roughly constant over the entire dose range of the statin. Here you're looking at the incremental effect of gemcabene on LDL percent change from baseline in the presence of different statin doses, in this case atorvastatin doses. So what you're looking at here on the top curve here is the dose response. You can see the x-axis is the gemcabene dose. So this is the gemcabene dose response for gemcabene monotherapy. And then as we go down here, these are what the dose response looks like as we increase the atorvastatin dose from 10, 40 to 80 milligrams per day. And what starts becoming apparent here is that the incremental benefit given by the gemcabene is decreasing considerably, so that while you actually have a pretty substantial benefit as you increase the gemcabene dose when there's no atorvastatin on board, when we get up to 80 milligrams, notice the curve is pretty flat. In other words, once you're taking that much atorvastatin, gemcabene offers very little additional benefit. This is viewing that in another way. Now what we're looking at is we're looking at atorvastatin dose response, again here, where in each case, the upper curve represents atorvastatin monotherapy as you go from no statin up to 80 milligrams per day. And the lower curve corresponds to, on the left-hand side, it's, I forget what dose of gemcabene was, but it was the sort of the main dose that they were targeting. I think it was actually, what was it? Excuse me while I take a peek. I think it was 600 milligrams, as I recall. And what you see here is that, excuse me, as the atorvastatin goes up, as we saw before, you get less and less incremental benefit from the gemcabene. Whereas with ezetimibe, in this case I believe that was 10 milligrams per day, the incremental benefit is roughly constant over the entire dose range. I'll even skip this table, is pretty much just redundant with the picture we just saw. But the net result here was to conclude that it performed unfavorably relative to ezetimibe, and the result was a decision to terminate the development program because it couldn't compete favorably with ezetimibe as combination therapy with statins. This is just a quote from the article where they said, in conclusion, the availability of the integrated model combined with the model visualization tool called DMX. So keep in mind this was done by Farsight at the time, so where they were pitching the software. Anyway, that led to a quick decision to stop the development of gemcabene. The model contributed significantly to the decision because it provided a quantitative comparison between gemcabene and ezetimibe when administered alone or in combination with a statin, and that those treatment options were not directly compared in any phase 2 trial. And by doing the integrated model across all the statins in combination with these, the predictions were more precise than they would have otherwise been. So that also increased the degree of certainty they had regarding their decision to stop development. Okay, let me take a look at another example. This is a more recent one, and talking about a longitudinal model-based meta-analysis in rheumatoid arthritis. In this case, the example here, in this example, model-based meta-analysis was used during the development of an agent called canakinumab to assess its efficacy relative to a number of marketed drugs. The model was developed to describe ACR20, which is a most commonly used measure of efficacy for rheumatoid arthritis response. So it was used to describe that ACR20 response over time for, you can see a big list of drugs here, abatacept, adalimumab, anakinra, certolizumab, I think I'm missing a vowel in here. I thought it was etanercept. Anyway, adalimumab, infliximab, rituximab. I'm not even going to keep pronouncing them all, but you can see a pretty sizable collection. And those results then were used to assess whether canakinumab was sufficiently efficacious to warrant further development. Actually, I don't know the entire story behind this, but I suspect it began as probably more of a dose-finding experiment, more than it was a proof of concept, but it became apparent that it was more of a proof of concept issue later on. They did the model-based meta-analysis based upon a collection of longitudinal data for all of these compounds, and this table just lists the number of studies for each one of those compounds, the corresponding number of patients that data represented, and so the data they're working with is fraction of patients achieving ACR20 and 37 double-blind controlled clinical trials and longitudinal data up to 54 weeks. Oops, jumped too far. This is just illustrates their modeling results here. So those are a little bit tiny. Hopefully in your handout you can see them a little more clearly. The y-axis here is the percent of patients that achieve ACR20, and the x-axis is the time in weeks, and it goes out to 54 weeks in this case, roughly a year. And you can see the model predictions shown here as curves with the shaded area around them. I don't recall how broad these were. I think these were probably like 90 or 95% prediction intervals around these. And you can see that compared to the observed data here. So these illustrates the model fits. And these plots now become more relevant to their decision making. These illustrate comparisons of canakinumab with two different compounds. I guess I did. So here we've got, actually I'm trying to decide where one puts the accent on etanercept. I'll just run with etanercept. Anyway, so we've got canakinumab versus etanercept and canakinumab versus adalimumab. And in both plots here, well, again, we're looking at percent of patients with ACR20 versus time. The lower curve on both of these represents the placebo outcome, and the upper curve represents the response to the comparator drug. So etanercept is up here, and on the right-hand curve, it's the same. It's the time course for adalimumab, and then stuck in the middle here, you can see are the model-based predictions for canakinumab. And you can see there is relatively little overlap in both cases with the time course for the comparator compound. So indicating that at least at the doses that they had selected for canakinumab, it resulted in an inferior response compared to those particular compounds. The end result then of this model-based meta-analysis exercise was then that there was a decision to not pursue the RA indication for that compound. And they stated, again, this is a quote from the article, that the integrated assessment of canakinumab data with the quantitative knowledge about current standard of care treatments supported that decision, given that the likelihood was low that canakinumab at current doses and regimen could provide incremental benefit to patients as compared with existing therapeutic options. So again, this is an example where the model-based meta-analysis was used to support a fairly key strategic decision. The next and the last example I'll go over today here is one where the model-based meta-analysis was used in a somewhat different way, and I might, in fact, to some degree, I suppose I may even be stretching the concept that many people would apply to the model-based meta-analysis, but that's really what it is. And in this case, it was used in the context of trial simulations to design a phase 2 dose-finding strategy. In this case, I've got to, I guess, protect the guilty or whatever here, so it's a blinded case in this instance. But we've got a case where there was a drug in phase 2. The idea was to design a phase 2 dose-finding strategy for this compound, and we'll call this compound drug X. And the phase 2 objective was to efficiently find a dose of drug X that is at least non-inferior to the standard of care, which we'll label as drug R, 10 milligrams per day. And it's non-inferior in the sense that it's with respect to both efficacy and safety. And we want to do that with sufficient certainty that we could risk a phase 3 program with only one dose level. The efficacy measure in here involved the decrease in the fraction of patients with a particular disease-related event. And the probable dose-limiting adverse event for this drug, again, involved an event count and the biological mechanism for that particular type of adverse event. It was really the same underlying mechanism as the one that drives the efficacy. The available information for drug X were clinical pharmacokinetic data from phase 1, and a preclinical biomarker that's thought to be predictive of clinical outcomes related to the mechanism of action for both efficacy and that dose-limiting adverse event. For other compounds, we were working with public source data on both the event counts for efficacy and the dose-limiting adverse events. In addition, we had some summary data on pharmacokinetics for those compounds. The basics, this is just a schematic for the modeling strategy that was used for this case that involved first constructing a model for the relationship between that preclinical predictor and the clinical outcomes. And to do that, we need an intermediate model here that predicts the relationships between preclinical and clinical outcomes. And in this case, that model is built based upon the combination of the preclinical data and the public source data for other compounds that worked by a comparable mechanism of action in here. Now, for the new compound, we did not have the key clinical outcomes of interest yet in any trial, so we had to use this relationship then plus the preclinical data to make inferences about the likely clinical outcomes for the new compound. And we used the model developed in that fashion to simulate clinical trials and to evaluate the performance characteristics of various clinical trial designs. In this case, we argued that the simulations led to more efficient designs that can find a non-inferior dose with high probability, or at least higher probability. And I won't go through, this is something we'll actually go through in more detail at a later point in the course, but just to give you a basic flavor here, one of the things that was compared was how the trial would do if you used fairly conventional statistical analyses that did not incorporate any prior information as part of it versus a trial design that used Bayesian modeling that incorporated that prior information. In the case where you used a conventional statistical analysis with no prior information, saw that the best performance results were seen with 7 log-spaced doses in our dose-finding study, and that only trials where we had substantially more than 1,000 people, only trials of that sort of scale offered sufficient certainty to consider risking a phase 3 program with only 1 dose level. Whereas if we incorporated the Bayesian modeling with prior information, it increased the probability of selecting a non-inferior dose, particularly in trials with smaller sample size. And in this was actually an adaptive design that had an adaptive pruning component, and it turned out that the use of prior information substantially improved the probability of making correct pruning decisions. And that combination really, that improved the performance quite a bit. So the end result is that, again, it led to more efficient designs. By leveraging prior information, it permitted a more efficient design and analysis of a phase 2 trial to select a dose for phase 3. It optimized the range, number, and spacing of doses. It optimized an adaptive pruning strategy for assigning patients to the most relevant doses, and enhanced characterization of dose response and therefore dose selection. The result was we ended up with a shorter, more informative phase 2 program, and I hear I say reduction by about 400 to 600 patients, which translates to roughly a 4 to 6 month time period at a typical recruitment rate, and the net result would then be a shorter time to market on the order of 4 months, and as I just commented, and potentially much greater shortening by avoiding an incorrect dose selection that could have led to a failed phase 3 program. And I think, that's about where I'm going to transition here. Let me again take a breather and see if there are any questions or comments.
对，这个连到电脑上就可以。
Hello?
这个连到电脑上不行吗？
连到电脑上以前。
Okay, well, if there are no questions, this is actually the point where I'm going to turn this over to Jonathan for the remainder of today's session. And as I think I mentioned earlier, we will have a lab session. It may be a somewhat abbreviated one this coming Monday. We won't have an assigned problem for that one, but it'd be a good time to just get you up to speed to our setup in terms of using the flash drive and the various components of the software that we're going to be using in the course. Maybe I can come up with an illustrative example just to give you a feel for how we'll be working with these tools. So I guess with no further ado, I'll turn it over to Jonathan. Of course, you know I'm dying to say, here's Johnny.
You could do that. It would make you Ed McMahon, I guess.
There we go.
Now we have to laugh. Yeah, right. Well, thanks, Bill. Hi, everybody. We're going to take the remainder of this session and probably the next, I don't know, 2 other sessions after that, 2, maybe 3 sessions, talking about some of the traditional methods for meta-analysis. And that's going to range from sort of fixed effects meta-analysis, which was very simple, very classical methods from probably developed in the 1950s, to random effects meta-analysis, including things like meta-regression and ways of explaining between study heterogeneity. And then we're going to get into the more modern but still traditional methods, modern methods that grew out of those traditional methods called network meta-analysis.
Apparently she said you were cutting out now and then, so I was going to plug you in if I...
Here.
Hopefully that doesn't disrupt the...
Okay. So we're gonna in the next few lectures, what we're gonna do is we're gonna span that kind of history of meta-analysis, primarily to give you a grounding in getting ready for the understanding the more complex model-based methods, and really to allow you to be able to critically evaluate the meta-analysis literature when you're reading them. So, you know, in that process, though, what we're going to do is we're going to start kind of slowly, and you know, in the very beginning, we're going to start very slowly. So let's just think about a situation where you might have had 5 data points, such as these shown here on the screen, and the question is, we want to get an estimate of the average or the mean of those data, and we can think about how we might approach that. We can see that these 5 data points, 3 of them are sort of clustered together there in the middle. There's one on the left that's a little bit lower. There's one on the right that's quite a bit higher. Think for a second for yourself how you might get an estimate of the average of those values, some measure of the middle of that distribution. So your first answer might be that we would just sort of take a simple average. If we have five data points, we'll just take the arithmetic mean of those. And in this case, that's identified here in this picture as X bar, and that falls somewhere here between the 3rd and the 4th point. But if I gave you a little bit more information, if I told you, for example, that some of the values were known more precisely, so for example, this value of minus 0.32 was known with a variance of 0.06, so that's known more precisely than say the value of that's minus 0.651, which has a variance of 0.932. We might be able to use that information in helping us get a more precise measure of the central tendency of these data. And the way that we could do that is by doing a weighted average of those 5, where the values where the weights are corresponding to the inverse of the variances of those values. So that the values that have a large variance, such as minus 0.651 and 0.946, which have large variances, are given relatively little weight, whereas the values that have small variances, like that minus 0.32, are given a relatively large amount of weight. In this context where we have the variances are differing, this is a more efficient estimator in the sense that the standard error of this estimator would be smaller than the standard error of that sort of simple mean estimate. And so what we need to, at a very, very high level conceptual idea, meta-analysis really is just a weighted average. So if you can imagine we were doing a meta-analysis of these 5 data points, we could think about there's two ways of approaching it. One is just to take this simple arithmetic mean. One way is to take this weighted average, where the weights are proportional to the inverse of the variances. And as we'll see over the course of the next lecture or two, the fixed effects meta-analysis estimate is essentially equivalent to that weighted average that we saw in the previous slide. And when we get to talking about the random effects meta-analysis, we'll see that the random effects meta-analysis lies somewhere between a simple average and that weighted average from the fixed effects meta-analysis. The random effects meta-analysis is in fact a weighted average, but it turns out that when there's a lot of between study variability, the weights are all become roughly the same. And so with equivalent weights, we essentially get what's equivalent to a simple arithmetic mean. And when there's very little between study variability, the weights become more like things here that are proportional to the inverse of the variance. And so it becomes more like that weighted average of that fixed effects estimate. So at its very simplest, just keep in mind that traditional methods of meta-analysis are really just very fancy ways of getting weighted averages. And you could even think about model-based meta-analysis in some sense as being a fancy way of doing weighted averages. And if you really wanted to stop there, we could just stop, and that could be the end of the course. And because I think that's the essential thing, at least for the traditional methods, that you want to take away. But we're not going to do that. We're going to, of course, forge ahead for the next 15 weeks or so. If you want a formal definition of meta-analysis, Sharon Leese Norman provided one in an article that she wrote in Statistics in Medicine almost 15 years ago. In that article, she defined a meta-analysis as a quantitative review and synthesis of results from related but independent studies. For the purposes of this course, we're going to distinguish between meta-analysis of aggregate data, which is an analysis based on summary statistics from trials or from arms within trials. And so those summary statistics could be things like a mean response or a proportion of responders, or they could be measures of treatment effect, things like a difference between two groups in a study, or say a difference between an active and control group, for example. Could be measures of effect on an odds ratio scale or a hazard ratio scale. But in any event, the meta-analysis there is of those summary level statistics from trials. And we'll contrast that with a meta-analysis that's based entirely on patient-level data, which might be something that you can imagine doing if you're putting together a new drug application and you have multiple trials from the same drug that you're trying to summarize at a high level the average effects of that drug at particular doses. We're not going to talk in this class much at all about combining purely patient-level data, although that is considered to be the gold standard when doing a meta-analysis. For the purposes of this course, we're going to focus on meta-analysis of aggregate data or a meta-analysis that combines aggregate data and patient-level data. As it turns out, when we're doing longitudinal models, which we will be getting to very soon after we get into the model-based meta-analysis portion of the course. You can imagine that we could apply either approach. We could apply, if we have longitudinal data, we can apply models at single time points or modeling the time course. In that case, if you have the patient level data, it's much better suited, particularly if we're going to talk about exploring effects of covariates. However, if, in the absence of having patient level data, using aggregate data will be better than nothing. It's important, though, to contrast a meta-analysis with what's in the literature called a systematic review. In general, systematic reviews are more qualitative summaries, whereas a meta-analysis is more of a quantitative summary. In both cases, they're related to a specific set of research objectives, which we'll talk more about in a minute. However, in the recent literature, I think the use of the term systematic review is kind of getting conflated with a meta-analysis. And so sometimes you'll see the term systematic review being applied to a literature search and an evaluation of that literature, and then a meta-analysis at the end. Historically, a systematic review is a more qualitative approach, but more recently that they often include this quantitative meta-analysis. There's some very good information in the literature about how to perform a systematic review and how to perform a meta-analysis. If you're not familiar with it already, the Cochrane collaboration provides their own guidelines. They have a handbook on performing systematic reviews and meta-analyses. And then in addition, there was a collective statement called the PRISMA statement that provides standards for reporting of systematic reviews and meta-analyses. I think later in the course we'll come back to the PRISMA statement and touch on some of the concepts about what's important to report in when you're publishing a meta-analysis, or when you're presenting them to perhaps a group internal to your company, some of the things that you need to be considering. If you're looking for references, I think for traditional meta-analyses, there's really two books that, in my opinion, stand out. One is published in 2009 by Bornstein, Hedges, Higgins, and Rothstein. There's also an excellent book by Ann Whitehead that was published now about over 10 years ago called Meta-Analysis of Controlled Clinical Trials. Both of these are excellent books and would provide a great complement to this course. If you aren't looking for a book but you want to read something shorter, the paper I referred to earlier by Sharon Lease Normand would be a good place to start. It's a review article in Statistics and Medicine from 1999. So what I'm going to start talking about next is the importance of thinking about your analysis before you do it. A lot of times we like to just sort of jump into sort of the technical aspects of modeling, but it's always a good idea to plan any sort of analysis, meta-analysis in particular. And I think you can really consider meta-analysis as being conceptually the same as conducting, running a clinical trial or conducting any sort of primary research. And so in keeping with that, we should always consider writing some sort of protocol or analysis plan or something that's going to state what the objectives of our research is. What questions do we want to answer with this modeling? That also then defines our inclusion and exclusion for the studies that we're going to be included as part of the analysis, and describe how we plan to analyze the data. And you can, I think, see the parallels there with running a clinical trial, in the sense that when we run a clinical trial, we always define the objectives of the clinical trial. We set out the inclusion-exclusion criteria for patients within that clinical trial, and then we talk about how we're going to analyze and summarize the data. And in the process of putting together that protocol for running a clinical trial, we do that with a multidisciplinary team that will include clinicians, statisticians, clinical pharmacologists, and operational scientists who help execute the clinical trial. In the context of doing a meta-analysis, we really should be thinking in the same broad sense that in performing that, we should build a multidisciplinary team that includes clinical subject matter experts, statisticians, clinical pharmacologists, and pharmacometricians, and information scientists to help with the literature search. The clinical subject matter expert may help us define the objectives of the research and what the inclusion-exclusion criteria should be for which studies to be pooling. So when you're thinking about your meta-analysis, just keep in mind the principles of running or designing a clinical trial. So in the context of this performing the meta-analysis or the systematic review, we can really think about there being 5 steps. The first is framing that question or coming up with the objectives of your analysis. The second is identifying the relevant literature, and that includes the step of what are the inclusion-exclusion criteria for the studies, as well as going out and retrieving the studies. The third step is then assessing the quality of those studies, so reading through each of the papers that you've got, ascertaining whether or not they meet your inclusion-exclusion criteria, and ascertaining the quality of the studies themselves. And then comes the summary of the evidence. That's our meta-analysis piece. And then there's an interpretation and reporting of the findings that comes out at the end. As for the analysis plan, as I stated before, you want to think about one or more well-formulated research questions, and I'll give a couple examples about what that might look like in just a minute. When you're thinking about inclusion-exclusion criteria, those are going to be things along the lines of what patient population would you like to include. So if you're doing a trial in a particular disease area, you'd want to think about whether you wanted to include only elderly patients or all patients, for example. You'd want to think also about the types of studies, whether you're including only controlled clinical trials, and if you are including controlled clinical trials, whether they have to be randomized. So this means, are you thinking about including observational data, for example? If you're working in oncology, do you include single-arm studies or not? If you're, in addition to those inclusion-exclusion criteria, thinking about what are the treatments or interventions that are of interest, and those may be comparators or controls within studies, or they may just be the experimental treatments in the clinical trials that you're pulling into your literature review. And finally, you want to think about the outcomes of interest and whether or not you have to have those outcomes in the papers, whether or not those outcomes have to be reported in order for you to include them in the cohort of studies that you'll be using for your meta-analysis. Some studies may report endpoints related to some of the research objectives, but not all of them, for example. And then in your analysis plan, also think about how you're going to be extracting the data and a short description about the analysis methods. And I didn't put it on here, but I think it's also important to consider how you're going to ascertain potential sources of bias. One of the important elements of the PRISMA statement is to, as you're reporting the results of the meta-analysis, is to think about where there are potential sources of bias and how you have attempted to address them either in your selection of studies or in your analysis methods. And those potential sources of bias can be related to how you've selected the studies. For example, did you only take things that were published in the peer-reviewed literature, or did you make efforts to find studies that were not published to try and prevent any sort of publication bias? But they also relate to potential biases within studies, and that might be related to things like how much dropout or discontinuation there are within a particular study. So let's look at 2 examples now of how we can think about stating some of the key elements of an analysis plan. And so I'm going to take 2 examples from the relatively recent literature. The first one here is a paper that was published in the Annals of Internal Medicine earlier this year on the comparative effectiveness of treatment for hepatitis C virus infection in adults. This was a work that was sponsored by the Agency for Healthcare Quality and Research, which is an agency in the United States that that funds some comparative effectiveness research. In this particular, for this particular article, this is a shorter synopsis of a much longer report that you can get at the AHRQ website. In this case, the authors had 4 objectives. They were interested in focusing in particular in patients with HCV infection. What is the comparative effectiveness of antiviral treatment in improving health outcomes? What's the comparative effectiveness on the rate of sustained virology response, or SVR rates? What are the comparative harms associated with antiviral treatment? And have improvements in SVR been shown to reduce the rates of adverse health outcomes? And in particular, there they focused on mortality. So you can see these are a really wide-ranging set of objectives looking at comparative effectiveness with intermediate outcomes on things like SVR rates. So that's an efficacy endpoint. Comparative outcomes in terms of potential harms that are associated. In this case, they focused on adverse events and serious adverse events. And then trying to connect whether improvements in SVR rates also reduce the rates of mortality. So in here, they specifically defined what their populations were, what the interventions of interest were, what the outcomes of interest were, and what kinds of studies they were including. So for the population, they were only interested in trials that recruited adults with HCV infection who have not had previous antiviral drug treatment. So for example, they were specifically excluding trials where they recruited patients who had failed antiviral drug treatment. The interventions of interest were pegylated interferon alpha-2A with ribavirin, pegylated interferon alpha-2B with ribavirin, and then at the time, the 2 protease inhibitors that they're looking at were telaprevir and boceprevir. And for those studies, they were either used in combination with a background of interferon ribavirin or as monotherapy. And then the outcomes of interest were the SVR rates. That was their primary measure of intermediate-term efficacy. Mortality from HCV, that was a measure of longer-term efficacy, as well as withdrawals due to adverse events and serious adverse events themselves. Of course, the studies now that they used were including clinical trials, but they also included the observational studies and systematic reviews. So essentially, all that they were excluding in terms of their set of studies were case studies and small case series. So that's one example where they clearly define their objectives, the populations, the interventions, the outcomes, and the types of studies. Here's another example that was published in 2006. And this was one that was published by the Cochrane collaboration. It's in their Cochrane database of systematic reviews. It was focusing here on the effects of chemotherapy and radiotherapy for inoperable advanced pancreatic cancer. The format of how they lay out their objectives and their populations and interventions is a little bit different than the AHRQ format. Here they give us the specific objective being to assess the effects of chemotherapy and radiotherapy on overall survival in people with inoperable advanced, including locally advanced and metastatic or relapsed disease. So we can see here, embedded in here is our interventions of interest, our chemotherapy or radiotherapy. Our outcome of interest is overall survival, and the patient population are patients with inoperable or advanced pancreatic cancer. The types of studies that they included were randomized controlled studies that were either single-blind or double-blind. So here they were not only that because of the potential issues with blinding and masking in chemotherapy and radiotherapy, they included single-blind studies. So single-blind or double-blind trials in which one of the intervention types, either chemotherapy or radiotherapy, was contrasted with either a placebo or another type of intervention. So we can see here they're specifically not including single-arm studies. They did their best to include both published and unpublished studies in their systematic review. So those are two examples in the published literature. I would hope that when any sort of example that you would see in the published literature should clearly define these kinds of objectives and patient populations and study populations of interest. And in general, this is a good thing always for us to do whenever we're at the outset of planning a meta-analysis, whether it's a model-based meta-analysis or just a simple one. So let's now drill down into each of these little elements in terms of the other steps of the 5-step process. In terms of performing the literature search, the important thing is to, once you've defined the inclusion-exclusion criteria for your studies of interest, is then to really rely on people who are trained to do these searches. There are lots of tools to do them, but in order to make sure we're casting the widest net possible and capturing all of the studies that meet those inclusion-exclusion criteria, we should really, we're going to be most successful when we rely on people who are trained to do that. And when you're reporting those, you should always define what bibliographic and abstract databases were searched. In particular, in addition to things like PubMed, whether you searched summary basis of approval documents from the FDA, whether you took results from clinicalstudyresults.org, for example, and other sources. And just as a little tip, if oftentimes you'll do a search, you'll come up with a paper or a list of papers, always review the references in those papers because you'll invariably find other relevant studies to include in your analysis. But ultimately, our goal here is to include all of the trials, both published and unpublished, that fit those inclusion-exclusion criteria. Once we do that, have performed that search and retrieve the literature, then the next important step is this critical appraisal of the studies. There's a number of reasons why this is important. Probably the simplest one is that as the analyst, then you get to understand those studies that are going into your meta-analysis. And that's vitally important that you understand each of those studies that are going into the meta-analysis, to understand the potential complexities and intricacies and subtleties that each of those trials have. Occasionally, we'll, in the process of performing a meta-analysis, give scores to each of the trials that we're including in the meta-analysis. One type of those scores are called Haddad scores. The original reference for the Haddad score is provided there at the bottom of the slide. These are scores that are on a scale of 5 points, a 1 being a poor quality, and a 5 or 0, I guess, being a very poor quality, 5 being the best quality, in terms of whether or not they describe the randomization of the study, whether or not they describe the blinding of the study, something about withdrawal or dropout, and how the method for generating the randomization. So you can see this score is some measure to try to assess potential biases that could be coming into the study. So things like, if the study wasn't randomized and the blinding wasn't very good, you can imagine that any sort of subjective assessments may be questionable. Having said that, whether or not they adequately describe how the randomization was generated maybe isn't that relevant to some of the analyses that we'll be doing. But this is a type of score that is often used in the meta-analysis literature. Occasionally, you'll see in the literature methods to try to include all types of studies, regardless of the Haddad score, and include and allow the weighting in the meta-analysis to depend on the Haddad score or on some measure of quality of the study. I don't know that we'll get to discuss those methods within the course of this course, but it might be a special topic we'll come back to at the end of the semester. But ultimately, this critical appraisal of studies is that first step in your assessment of potential sources of bias in the studies and across the studies. And this will really form the basis for that evaluation in the reporting of your results. Of course, once we've got the papers, then the next step is to extract the data. Ideally, you'd have some standardized database formats to use for that, for the data retrieval, and then processes for utilizing those databases. And of course, before you do any extracting of your data, you want to think about what you're going to be modeling to make sure that you're extracting the right kinds of data. If you're in a situation where you're not actually doing any of the data extraction yourself and you're just on the receiving end of a data set, then you may want to be thinking about the quality of the data that you're receiving and perhaps doing a little bit of a evaluation, comparing one or two studies to in the database to the source data themselves. So there's this data extraction piece itself for you may or may not be particularly relevant. When you're thinking about, but if you are doing the data extraction, you're thinking about what endpoints to extract, just keep in mind your modeling objectives. And keep in mind that while you may be modeling, you may be choosing to model the response outcome itself, or you may be modeling directly the treatment effects, so for example, a difference from placebo. Or you may be modeling the treatment response and then presenting things in terms of treatment effects. So if you plan to be modeling the response endpoints, then you want to be extracting the response endpoints. If you plan to be modeling the treatment effect endpoints, make sure you've extracted those. So the kinds of treatment, if you have binary data, your response rates might be, or the endpoint might be a response rate, a proportion of patients responding, whereas a measure of effect could be a risk difference or an odds ratio. For continuous data, that would, contrasting observed means versus the difference between groups in means. And if it's time to event data, the endpoint data itself might be a median time to event, or it may be summary values of survival rates at particular time points that might have been extracted, for example, from a Kaplan-Meier plot. And just contrast that with a measure of effect, like something like a hazard ratio. Okay. So after you've done the data extraction, then the next step is summarizing the evidence and interpreting the results, and that's really going to be the focus of the rest of the course. So we're not going to have a specific slide on that topic. I'll stop there for a second to take any questions that anyone might have on planning a meta-analysis. The next step we're going to move into is just thinking about specific, the first steps in conceptualizing a simple traditional meta-analysis. So if you have any questions, the best way to do is probably just to type them into the chat box, or actually here I'll unmute you guys if you want to ask any questions. Okay, well, I guess there are no questions. We will then go ahead and turn next to our to this notion of traditional, simple traditional meta-analyses. So when we're thinking about a meta-analysis, we really need to think about three potential sources of variation. So at the simplest level, we have the sampling error. At the next level, we have some within study or some study level characteristics that'll give us some variability, as well as sort of any additional between study variation. So that sampling error, you can think of, that bigger studies are going to provide more precise estimates, smaller standard errors than small studies. And so a big study should give us an estimate that's closer to the truth than a smaller study. In terms of study characteristics, we're going to think about those as kind of dividing up all of the between study variability in terms of elements that we can describe through covariates or study level characteristics, And when we get to meta-regression, that's what we'll be doing, as well as sort of all of the remaining unexplained variability in treatment effects across studies. So we're allowing for the fact that there may be some between study variability in what the treatment effect is, and then given some sort of true treatment effect for a study, there'll be some additional sampling variability. That just comes through the natural process of aggregating the data. When we're performing these traditional meta-analyses, we can really think about there being two broad classes of models. The first that we'll start with are called fixed effects models. And the basic idea there is that each study is measuring the same underlying parameter, so the same underlying treatment effect, for example, and that there's no between-study variation in that treatment effect, and that may be after we've accounted for covariate effects. But in the simplest conceptualization, we will just say that there's no between-study variation in treatment effect. And that the only source of variation between studies is that sampling error. In the random effect conceptualization of the model, we say that each study has its own different but related treatment effect, and that those are related in a hierarchical fashion in the same way that we do in terms of multi-level or PK/PD models, where we'll have a study level effect, and then conditional on that study level effect, we'll observe something within a particular study. And you could imagine that if we have included covariates in the model in this random effect model, if we still need to have the random effects, that's simply telling us that there's some additional unexplained between study variability in addition to the sampling error. So if we want to think about that graphically, this is a figure from the paper by Sharon Lease Norman. Imagine that the fixed effects model is essentially saying that if we had 5 trials of varying size, all of the trials are the distribution for the sampling distribution of that mean are all coming from the same different distributions, but they're all centered on the same value, just denoted by this line here of theta. And the only reason that the variability in those studies and the distribution of the data from those studies is different is because of the sample sizes are different. So this curve here that's on top, that is the widest, is coming from a study where there's more sampling error, perhaps because it's a smaller study. And the study here that's the closest in, that is the narrowest distribution, is coming from perhaps the largest of the 5 studies. But in any event, all of these studies are centered on the same common effect. In contrast, the random effects model says that each study has its own particular average effect, and that those average effects themselves come from a distribution, that there's a sort of a population of these study level effects. And that then conditional on that study level effect, each study is centered on that study level mean value, and then there's some additional sampling variability around that. And so that's denoted here by this skinny little distribution here is a study that has a lot of information, or perhaps a very big study, and these 2 studies over here on the right, which have wider distributions, are coming from smaller studies where there's more sampling error. It's sort of the same kind of model that we would have when we're thinking about doing any sort of population analysis when we have patient level data. So for the fixed effects analysis, let's start with that. That's the simplest. And I'll just start by defining some notation. And I'm going to distinguish between using Y's for an observed summary data that's kind of the type that's like a response, so like a response rate or a change from baseline score. And I'm going to contrast that with, I'll use a theta hat to denote things that are treatment effects. So in the subsequent slides, if I use something like yi1 and yi2, those would denote things like the response rate in the experimental group or the response rate in the control group within the ICE study. Or if it's, and that might be for a study where the outcome is a binary outcome. If we have a continuous outcome, YI1 and YI2 might denote things like the mean change from baseline response in the experimental and control groups in the ICE study. And we'll just contrast that with the notation that I'll use for the treatment effects for the ICE study. I'll denote those by theta hat I, and that could be something like the log odds ratio comparing the experimental and control groups if we have a binary outcome, or the mean difference in change from baseline response if we have that corresponding continuous outcome. So the Y's are things like averages or response rates, and the theta hats are things like are differences or between groups. So what the simple fixed effects model says is let's just suppose that we have R independent studies, and they each compare the same treatment to a control. So this may be drug X versus placebo, for example. And theta hat I will be our estimate of that difference between the two treatments. And theta is going to denote our measure of the treatment difference, to the average measure of effect. In that case, then the general fixed effects model simply says that the theta hat I's are equal to the average effect theta plus some error term epsilon I, and that the expected value of the epsilon I is 0, and that the variance of epsilon I is psi I squared. And that's it. That's the model. Almost always in meta-analysis, we'll assume that we know the variance, or in this traditional meta-analysis, we'll assume that we know the variance of epsilon. So that is, we'll treat those psi's as known and equal to the estimated variance of the theta hats. So that is, if reported in the literature, you may have a measure of treatment effect, and then oftentimes you'll get a measure of its standard error, and that standard error here I've denoted by SI. And so SI squared then is the variance of that measure of treatment effect. So then what this fixed effects model essentially is telling us is that those theta I's, those treatment effects in the ICE study, are normally distributed with a mean of theta and a variance of SI squared, or SI squared is known. So the only unknown parameter here is theta. So you may see that if you're paying close attention that I've kind of snuck in a little assumption here on this slide that wasn't on the previous slide. I'll give you a second to think about what that was. Here, this is the slide where we've defined kind of the most general fixed effects model, and this is where I've defined kind of a slightly more nuanced version of that. And in this case, the assumption that I've imposed here is this assumption of normality. Strictly speaking, in a fixed effects meta-analysis, we don't have to make any assumption of normality. We can simply define this model here, the theta hat I is equal to theta plus epsilon I. The variances of epsilon I are known, and that expected values are equal to 0. But we'll go ahead and make this additional assumption about normality, and maybe think about that a little bit as we move ahead. So it turns out that if the variance of the theta hats is known, and that if it follows a normal distribution, then we can show that the maximum likelihood estimator for theta, I'll denote that as theta hat FE for fixed effect, is equal to the sum of a weighting term, I'll call that WI, times theta hat I, that's our data, divided by the sum of the weights. And in this case, the weights for each study is 1 over SI squared, 1 over the variance of that estimated treatment effect. So this is, if you remember back to when the very beginning, I talked about that weighted average where the weights are equal to the inverse of the variance. That's exactly what this at that gradient. That'll be our maximum likelihood estimator, times the sum of 1 over SI squared. So that's just a simple rearrangement of this middle equation here. And then solving for theta hat Fe, we get that theta hat Fe is the sum of theta hat I over SI squared divided by the 1 over SI squared, or the sum of 1 over SI squareds. And we can just re-express that as the same fixed effect estimator equation we had in the previous slide, where the Wi's are the 1 over SI squareds.
Hey, Jonathan.
Did someone have a question?
Yeah, it's me.
Yeah.
It's Joe.
Oh.
I want to let you know that you muted yourself. I'm not sure how long for, because I just came back and saw all the questions saying you'd muted. If someone can put in the question box, how long was he muted for?
From the start of the MLE slide. That's funny. So I didn't
From the start of the MLE slide. So you probably want to start over on that.
Okay, I didn't touch anything on my end, so I'm not sure what happened.
Did you hit the mute all button?
No. Let me see here. So, can you guys hear me now?
I unmuted you. You were muted.
All right.
Yeah.
I'm not sure how that happened. Well, my apologies. We'll just start back with the MLE slide and then go from there. So, okay. So, thanks, Joe. So to find the maximum likelihood estimator, what we need to do is write out the likelihood or the log likelihood as a function of our parameter of interest here. That's theta. And so in this case, because we have assumed a normal distribution, our likelihood is going to be the likelihood for a normal distribution. So the likelihood of our data theta I, given our parameter theta. In this case, actually, the SIs are also data. So the log likelihood from a normal distribution here is the sum over the I studies of minus a half times the log of the variance, log of SI squared, minus 1/2 times the theta hat I minus parameter theta squared over SI squared. So to find the maximum of that log likelihood as a function of theta, we'll take the derivative with respect to theta. This first term here doesn't depend on theta, so that drops out. And the second term, when we take the derivative with respect to theta, turns out to be theta hat I minus theta over SI squared. And then that's being summed over, again, over the R observations we have. And then to find where the maximum, or potentially minimum, but in this case maximum of that function occurs, we'll set the derivative equal to 0 and solve for theta. And so in that case, we have simple re-expressing this. We get the sum of the across the data of theta I over SI squared equals the theta hat Fe. I'll use that to denote the value of theta at the maximum value, times the sum from I equals 1 to R over 1 over times 1 over SI squared. And then rearranging terms and solving for theta hat Fe gives us the expression that theta hat Fe is equal to the sum of the theta hat I over SI squared divided by the sum of 1 over SI squared. And we can just, if we stare at that for a little bit, we recognize that that's our fixed effects estimator from before, where we've just denoted the Wi's as being 1 over SI squared. And so that's just a simple, quick derivation for this simple model to demonstrate that fixed effects estimator is in fact a maximum likelihood estimator, and then because of that has all of the nice properties of a maximum likelihood estimator. In the course of the course, we're going to be getting into much more Bayesian analysis, and you can imagine that you could fit a Bayesian version of this fixed effects model. And for that particular model, we need, for any Bayesian modeling, we need the likelihood, and we need prior distributions. And for this particular very simplistic case, you can imagine we might use a normal prior distribution for theta. So I'll imagine that we're doing this particular analysis where, I see my first typo here, this should be theta hat I, given the parameter theta is normally distributed with mean theta and variance SI squared. So this is just the same fixed effects model we had before. But now we've added in an extra layer where we're putting a prior distribution on theta. And in this example, I'll just assume that the prior for theta follows a normal distribution with mean mu and variance sigma squared. And in this case, it turns out that the prior distribution is a conjugate prior, so that we can actually derive what the posterior distribution is in closed form. And it turns out that for this combination of this normal model, that then our posterior distribution for theta conditional on the data, so that's our theta hats and our s's, is also a normal distribution with the mean of the note by mu sub post and a variance sigma squared post. We'll start with the variance. The variance sigma squared post is just the sum of the weights plus sigma 1 over sigma squared, that whole quantity inverse. And the posterior mean is the sum of the weights times the observed differences plus mu over sigma squared divided by the sum of the weights plus 1 over sigma squared. We can express this in closed form, and we can, if we wanted to do a simple Bayesian analysis in this context, we could simply write out what the posterior distribution was. Of course, if we are thinking about doing a Bayesian analysis where we had a relatively non-informative prior, and by non-informative here, what I mean is that sigma squared is large or very large relative to both mu and to these SI squareds, or to the, to the sum of the weights, to the sum of the 1 over the SI squareds. It turns out that we can show pretty easily that the Bayesian analysis is going to be very closely approximated by that simple fixed effects classical analysis, in that the posterior mean will be roughly equivalent to the fixed effects estimator, fixed effects maximum likelihood estimator, and the posterior variance will be essentially equivalent to the formula we had for the variance of the fixed effects estimator before. So just to see that, let's come back to this slide and look at these two expressions. So if 1 over sigma squared is small relative to the sum of the weights, then this term here is roughly equivalent to the sum of the weights, which is the variance of that fixed effects estimator. And if in addition sigma squared is large relative to mu, this term here in the numerator for the posterior mean is close to 0. And again, because the 1 over sigma squared is small relative to the sum of the weights, this whole term for the posterior mean can reduce to something that's very close to the sum of the weights times theta I divided by the sum of the weights, which is our classical fixed effects meta-analysis estimator. So if we're simply doing a conjugate Bayesian analysis like this with non-informative priors, you could almost argue that you can do the classical analysis and use the estimates from that in a Bayesian manner. Very similar to what you'd get in many other contexts where non-informative priors lead you to inferences that are very similar to maximum likelihood inference. Of course, if we don't want to do conjugate Bayesian analysis, and when we get into more complex models, we'll need to use other tools. In particular, for this course, we'll focus on using WinBUGS. Let me just take a quick look and see if we're coming to a place where, I think what we'll do is we'll go through probably one more slide here, and that'll get us to a place where we're at a good stopping spot for the day. Oftentimes when we're performing a meta-analysis, we may either not have the standard errors reported, or we may want to do something that's simpler, I'm sorry, I'm getting off track here. I'm jumping ahead in my thought process of what we're going to be covering. So in everything that I was discussing up to now, I've been talking about theta hat I, where theta hat I, if we come back here, was the estimate of a treatment effect. Oftentimes we may be interested in simply modeling what is the average response rate or what is the average change from baseline in a particular treatment group. So focusing not on a difference of treatment groups, but on a treatment response itself. And the question often comes up, can I use the same methodology if I'm working with a mean response rather than a treatment effect? And the answer is yes. In this case, though, so our model, rather than being that the theta hat I's come from a normal distribution that are centered at theta with a variance SI squared, we'll say that our response values come from a normal distribution with a mean theta and a variance SI squared. And then under those assumptions, our fixed effect estimator is still the same weighted average of the yi's now, where weights are 1 over the variance of those yi's. In this case, remember, yi itself is a mean value. So you can imagine that if yi is itself a sample mean, and we assume that across the studies, between subject variability, so now this is the variability in the response at the individual level is the same, and we'll just denote that by sigma squared, so that the standard error of that mean response is sigma over square root of n. Or another way of saying that is that the variance of the sampling variance of that mean is sigma squared over n. Then we could, those weights become 1 over SI squared turns into 1 over the quantity sigma squared over Ni squared, which it becomes Ni squared over sigma squared. So our weights are Ni over sigma squared. And then if we were to plug those values into our equation for the fixed effects estimator, we'd see that that constant sigma squared cancels in the numerator and denominator, and what we're left with is simply an estimator that's based only on sample size, which is in that estimator is specifically the sum of the ni's times the means divided by the sum of the ni's. So with mean response values, if under this particular assumption that the variance is constant across all of the studies, our estimated average simply becomes something that's based on sample size rather than on standard error of the estimates. I think with that, it's probably a good place to stop for the day. Let me see. I'm happy to take any questions. I'll let you type those into your question box or into the chat box. Okay, well, if there aren't any questions, then I think what we'll do is we'll stop here for the day, and we'll pick up next week with talking about what we can do when we don't actually get standard errors reported, and then that'll carry us in through probably some examples and random effects meta-analysis models. So thanks a lot, and if you have questions that come up over the course of the week, feel free to post them to the training site, and Bill or I will answer them. Thanks, and we'll talk to everybody on Monday.